repliQa Hi-Fi Tough Mix, Best Practices for Optimal PCR Amplification. In this video, we will describe key considerations to ensure successful amplification using repliQa Hi-Fi Tough Mix, a high-fidelity master mix for PCR. Since its invention in 1983, the polymerase chain reaction, or PCR, has driven significant advancements in scientific research and healthcare. Much of this success can be attributed to the evolution of Taq DNA polymerase. Key innovations, such as the incorporation of hot start antibodies and the development of high-fidelity DNA polymerases, have enabled faster, more reliable, and more accurate PCR amplification. However, other major applications have emerged which require specialized DNA polymerases with unique capabilities. These include next-generation sequencing, single nucleotide polymorphism detection, and whole genome amplification. Key attributes of DNA polymerases include specificity, thermostability, fidelity, processivity, long-range capability, inhibitor tolerance, and the ability to amplify high GC targets. However, most polymerases excel at only a few of these traits. To address this, QuantaBio developed repliQa Hi-Fi Tough Mix, which delivers on all these attributes and more. repliQa Hi-Fi Tough Mix is an advanced, next-generation PCR master mix offering 90 times higher fidelity compared to wild-type Taq. It's incredibly fast, with extension times as short as 1 to 10 seconds per kilobase. This highly sensitive hot start polymerase also has long-range capabilities, enabling the amplification of fragments up to 24 kilobase, even with varying GC content. Additionally, it incorporates QuantaBio's industry-leading tough mix technology, making it highly tolerant to a wide range of PCR inhibitors. Choosing the right cycling conditions is crucial for success and depends on the melting temperatures, or TM, of the primers. To determine the appropriate TM for use with repliQa, we recommend using the online program Primer 3 Plus with the check primer function using the default settings. For primers with a TM of 63 degrees Celsius or higher, a two-step PCR cycling program is recommended. For lower TM values, a three-step cycling program is preferred, where the calculated TM can be used as the annealing temperature. Running an annealing temperature gradient is recommended for best results. To determine the importance of primer TM in amplification using repliQa, we amplified a roughly 1 kb target from E. coli DNA, with primer sets having melting temperatures ranging from 52 to 68 degrees Celsius, using both two-step and three-step cycling programs. The results showed that repliQa Hi-Fi Tough Mix performed better with a two-step PCR cycling program when the primer TM was above 63 Celsius. In contrast, the three-step cycling program successfully amplified the 1 kb targets regardless of the primer TM. These findings demonstrate that for primer TM values below 63 degrees Celsius, a three-step cycling program is necessary to achieve successful amplification. Another area where repliQa Hi-Fi Tough Mix excels is in long-range amplification. This tape station electrophoresis confirmed successful amplification of targets ranging from 250 base pair to 20 kilobase from E. coli DNA. This demonstrates its versatility as an amplification reagent, capable of handling both small targets and long-range amplification without compromising PCR yield. repliQa Hi-Fi Tough Mix demonstrates robust performance in amplifying targets with a wide range of GC content. To test this, we used targets of different length for 3 bacterial species with varied GC content: F. nucleatum with 27% GC content, E. coli with 50%, and B. pertussis with 67%. The results shown here show that repliQa successfully amplified targets of varying sizes across all 3 species, showcasing its efficiency in handling different GC-rich sequences. repliQa can also be used to amplify libraries for whole genome sequencing. In order to test this, we compared repliQa and Spark Hi-Fi PCR master mix to amplify libraries that were prepared using the Spark DNA Frag and Library Prep Kit. Inputs of 10 nanogram and 100 nanogram of mixed bacterial genomic DNA, or MBGD, were used. In this experiment, a 1-second extension was used for repliQa, whereas 30 seconds was used with Spark Hi-Fi. Similar library yields were generated with both master mixes, even with only 10 nanograms input, suggesting that repliQa Hi-Fi Tough Mix produced high library yield with only 1 second of extension time. Both Spark Hi-Fi PCR Master Mix and repliQa Hi-Fi Tough Mix showed low duplication rates regardless of the DNA input amount or the number of cycles. Finally, the genome coverage across the range of GC contents was identical for both master mixes, showing that Spark Hi-Fi PCR Master Mix and repliQa Hi-Fi Tough Mix have little bias towards different GC content. repliQa was further recognized for its utility in NGS applications by the Quail team at the Wellcome Sanger Institute in the UK. In a recent paper, this team identified repliQa as the best polymerase for both short and long read sequencing, outperforming over 20 other polymerases tested. Please refer to the paper entitled Identifying the Best PCR Enzyme for Library Amplification in NGS for more information. In this summary video, we have shown you how to achieve successful PCR amplification with repliQa Hi-Fi Tough Mix following recommended guidelines. This master mix stands out due to its unique features, including inhibitor tolerance, robust long-range amplification capabilities, compatibility with a wide range of GC content, and its remarkable speed. repliQa Hi-Fi Tough Mix is a versatile, high-fidelity master mix with high performance for standard endpoint PCR, as well as library amplification and other sensitive sequencing applications. For more information and detailed protocols, please refer to the application note entitled repliQa Hi-Fi Tough Mix, Best Practices for Optimal PCR Amplification, or visit us on our website.